Hey, so all welcome back to my YouTube channel. Today we will discuss about the Krebs cycle. So before beginning with Krebs cycle, there were different names given to this cycle. It is also called as TCA cycle, or it is also called as citric acid cycle. Citric acid cycle. Like the glycolysis, it was done in the case of cytoplasm, while the Krebs cycle is done in mitochondria. But you should remember that the cells which lack the mitochondria don't have any Krebs cycle. So what are those cells that don't have mitochondria? Basically they are RBC, they are cornea, and they are the lenses. Okay, they don't have mitochondria. If they don't have mitochondria in them, so in these three structures there will be absence of Krebs cycle. Okay, these were the other name, that is the TCA cycle and the citric acid cycle. This cycle undertakes in the place of mitochondria, and this cycle is absent in the structures which lack the presence of mitochondria. Like it can either be RBC, it can either be cornea, or it can either be our islands. So let's begin with citric acid cycle. First of all, you should remember that it begins with acetyl CoA. Basically, it is a 2-carbon compound, and you should remember that one molecule of acetyl coenzyme can give 10 ATP. Let me write down here. One molecule of acetyl CoA can give 10 ATP. Okay. So basically, here what will happen in the classification, acetyl coenzyme A will convert into citrate. I will firstly mention about the cycle and then I will tell you about the enzymes so as to make it simpler for all of you to remember. So basically acetyl CoA will convert into citrate. Please follow the words. Like here there is C, so next word will be citrate. Now here the word comes Ci, so next word will be cis aconitate. So here Is is coming, so next word will be isocitrate. Here the word is O, so next word will be oxalosuccinate. Okay, you should remember, first of all, there is a presence of acetyl CoA. In the acetyl CoA we have the word C, so next will be citrate. While in the case of citrate, first two letters are C and I, then next word will be cis aconitate. From the cis aconitate, take the term Is. The next word will be isocitrate. From the isocitrate, take the word O. So the next word will be oxalosuccinate. Okay. So next we have the alpha ketoglutarate. Okay. From the alpha ketoglutarate we have succinyl CoA. From succinyl CoA we have succinate. From the succinate we have fumarate. From the fumarate we have malate, and from the malate we have oxaloacetic acid, and then the acetyl coenzyme A. Half part of it is very easy that you should remember. First of all, there is acetyl CoA. Then from the acetyl CoA we have the citrate. From the citrate, learn the two terms Ci. Then we will have cis aconitate. From the cis aconitate, learn the two terms Is. Then we will have isocitrate. From the isocitrate, we will have oxalosuccinate. From the oxalosuccinate, you have to remember about the alpha ketoglutarate. If you have remembered the alpha ketoglutarate, then it will be easier for you to remember the steps afterwards this. From the alpha ketoglutarate, we will have succinyl CoA. From succinyl CoA, we will have succinate. From succinate, we will have the fumarate. From the fumarate, we will have the malate. And from the malate, we will have the oxaloacetic acid. Okay. So now what's the most important thing is the presence of enzyme. Basically, acetyl coenzyme A is converted to citrate with the help of enzyme that is called as citrate synthase. that is citrate synthase. From the citrate to cis aconitate, we have the enzyme that is called aconitase. Okay. From cis aconitate to the isocitrate, we have also the aconitase enzyme. From isocitrate to oxalosuccinate, we have isocitrate dehydrogenase. Okay. From oxalosuccinate to alpha ketoglutarate, we also have the same enzyme, that is isocitrate dehydrogenase. And from alpha ketoglutarate to succinyl coenzyme A, we have the enzyme that is succinate thiokinase. And from succinyl CoA to succinate, we have succinate dehydrogenase. From succinate to fumarate, sorry, I have mentioned it wrong. Wait for a moment. From alpha ketoglutarate to succinyl coenzyme A, as per the name, it is alpha ketoglutarate dehydrogenase. Okay. From succinyl coenzyme A to succinate, we have succinate thiokinase. Okay. From succinate to that of the fumarate, we have succinate dehydrogenase. From fumarate to that of malate, we have fumarase. And from malate to oxaloacetic acid, we have malate dehydrogenase. Dehydrogenase. Okay. So you should remember. Let's revise it again. First of all, acetyl CoA will convert into citrate with the help of enzyme citrate synthase. Then citrate will convert into cis aconitate with the help of enzyme aconitase. Then cis aconitate will convert into isocitrate with the help of enzyme aconitase. Then isocitrate will convert into oxalosuccinate with the help of enzyme isocitrate dehydrogenase. Oxalosuccinate will convert into alpha ketoglutarate with the help of enzyme isocitrate dehydrogenase. Alpha ketoglutarate will convert into succinyl CoA with the help of enzyme alpha ketoglutarate dehydrogenase. Then succinyl CoA will convert into succinate with the help of enzyme succinate thiokinase. And succinate will convert into enzyme the fumarate with the help of succinate dehydrogenase. Then fumarate will convert into malate with the help of enzyme fumarase. And malate will convert into oxaloacetic acid with the help of enzyme malate dehydrogenase. First enzyme you have to remember, that is citrate synthase. Next enzyme you can remember easily, like when the citrate is converted to cis aconitate, and the last word is aconitate. So from citrate to isocitrate, and from isocitrate to from cis aconitate to isocitrate, we have the same enzyme, that is aconitase. And till alpha ketoglutarate, we have also the same enzyme, that is isocitrate dehydrogenase. Then the enzyme will change. Okay, please remember this very carefully. Next, importantly, we are going to discuss some important things in this. Let me change the pencil color. Okay, you should remember that this oxaloacetic acid is basically the first product that is used in citric acid cycle. Okay, and it is basically this is basically the first product to be used and the last to be consumed, last to be ended. Okay, so basically this was about oxaloacetic acid. So next are there some steps that you should remember, like I have taught you about that during the conversion of succinyl CoA to succinate, there is an enzyme called succinate thiokinase. During this step there will be the presence of 2 of the ATP. Okay, and this step is called as substrate level phosphorylation. Okay, and during the conversion of succinate to fumarate, we have an enzyme that is called succinate dehydrogenase. Here we will have the presence of 3 ATP. Okay, and presence of 2 FADH2 molecules. Okay, and from conversion of malate to that of the oxaloacetic acid, here will be the production of 5 ATPs. Most importantly, when there will be the conversion of oxalosuccinate to alpha ketoglutarate, there will be also the presence of 5 ATPs, and from alpha ketoglutarate to succinyl coenzyme, there will be also the production of 5 ATPs. Okay, so this you should remember. Let me tell you about the procedure of ATP again. From oxalosuccinate to alpha ketoglutarate and from alpha ketoglutarate to succinyl CoA, there will be the generation of 5 ATP in both process. And the succinate thiokinase, in the succinate thiokinase we will have the generation of 2 ATP, while in the case of succinate dehydrogenase we will have the generation of 3 ATP and 2 molecules of FADH2, while during the conversion of malate to oxaloacetic acid we will have again generation of 5 amount of ATP. So if we calculate all the steps where the energy are produced, how we can calculate is that this 5 ATP, 5 ATP, this 2 ATP, this 3 ATP and 5 ATP. Let's make a total 5 plus 5 plus 2 plus 3 plus 5. This basically constitutes of 20 ATP. Okay. So these steps where the ATPs are produced, they are basically the energy producing steps in the case of Kreb cycle. Okay, that you should remember. And another thing that you should remember is that this comes very often in your this paper, so you should remember it very well. Okay, so basically these steps are the energy producing steps. So let's revise this Kreb cycle again. First of all, there will be acetyl CoA that is converting to citrate with the help of an enzyme, that is citrate synthase. Then our citrate will convert into cis aconitate with the help of enzyme aconitase. Then cis aconitate will convert into isocitrate with the help of an enzyme, that is aconitase. Then isocitrate will convert into oxalosuccinate with the help of enzyme isocitrate dehydrogenase. Then oxalosuccinate will convert into alpha ketoglutarate with the same enzyme, that is isocitrate dehydrogenase. Then alpha ketoglutarate will convert into succinyl CoA with the help of enzyme alpha ketoglutarate dehydrogenase. Then succinyl CoA will convert into succinate with the help of enzyme, that is succinate thiokinase, that is the substrate level of phosphorylation involving our Kreb cycle. Then succinyl CoA will convert into succinate, and then succinate will convert into fumarate with the help of enzyme, that is succinate dehydrogenase. And then fumarate will convert into malate with the help of enzyme fumarase, and malate will convert into oxaloacetic acid with the help of enzyme, that is malate dehydrogenase. So if you remember these terms, then you can easily follow the next step. Okay, like if you remember the citrate, the next step you can remember it the cis aconitate. If you remember the cis aconitate, the next step will be isocitrate. You can remember, like here the word C, so next word will come from that of the C, that is called the citrate. From okay, so next word here will come from cis aconitate. Here next word will come from isocitrate, and here the next word will come from oxalosuccinate. Okay, please remember this is the substrate level of phosphorylation. Okay, so this was all about our Kreb cycle that you should remember. Please do like, share and subscribe to my channel if you find this video informative, and please share with your friends. Thank you for watching.